Hello, this is the first episode of amino acid and protein, and in the first episode, we're going to talk about amino acids. This would be the only amino acids structure breakdown you need for MCAT Biochemistry, so hang on tight. We have 20 amino acids, we have 5 different categories, we have 4 different groups hanging off one tiny carbon, and somehow you're supposed to memorize all of it, recognize it instantly on a passage, and figure out how it behaves at 3 different pH values, which are the neutral, acidic, and basic. I know it sounds frustrating, but the good thing is, amino acids aren't 20 random puzzle pieces. They're one repeating pattern with a single changing part. And once you see the pattern, the memorization problem mostly disappears. So, because you're not memorizing 20 molecules anymore, you're memorizing one shape and then just swapping out a side chain. Before diving to the amino acid, it is important to know that amino acids are the building blocks for proteins. So you have to know the structure. So you have to understand amino acids in order to understand what kind of function those proteins could have with the amino acids that are provided to them. So we're going to dive into just like looking at just having a general view of the proteins and before amino acids. We're going to talk about proteins more in the next chapter. So the whole chapter is talking about the amino acids, but if I want to talk about it specifically, it's going to be in the next chapter. So let's talk about the protein functions. We have 4 major functions of protein in a cell. They can act as catalysis, they can do transport, they can do structure. There are more to it, but these are the major ones. And they can do motion. As for the catalysis, we can use DNA polymerase as an example. DNA polymerases are those that help in a DNA replication. As for the transport, we can talk about hemoglobin, which is actually the structure that we have right here. And this is the active site of the hemoglobin, which we would talk about in the protein chapter. As for structure, we can mention collagen, which is like basically a connective tissue. And as for motion, we can talk about actin and myosin, which are mostly in the biochemistry, but which I haven't made yet, but I would do it soon. The protein right here is called luciferin, and it's basically in fireflies, and this is how fireflies produce light. And what it does is just basically the ATP would hydrolyzes to AMP, and then it would attach to it, and then the removal of AMP causes the production of light. But it's not important. We're going to talk about it later. So now let's talk about amino acid themselves. We have one structure and one variable group. Every single amino acid, all 20 of them, is built on the same exact skeleton. This is basically the skeleton that we are talking about. This is the skeleton that we are talking about, this one right here. So we're going to draw it first. We have a central carbon. We have a central carbon. This one. I'm going to put a starter. We have a hydrogen group. We're going to have a carboxyl group. We're going to have an amine group. And then here's the side chain. This is called side chain, or they call it variable group, or sometimes they call it the R group. Doesn't matter. This is basically what we're going to talk about. And every amino acid, all of them have the same exact structure except this R group right here. So now we know about the structure of amino acids. We're going to talk about the 5 classes of amino acid, sorting by their side chain. And it is because since the R group is the only variable, it makes sense that we sort amino acids by what that R group looks like. Each of those groups that we talk about, classes that we talk about, each of them tells you something about how that amino acid will behave in a protein. The first group is the nonpolar amino acids. We have 7 of them. And as we said before, amino acid have the same structure. We're going to start by glycine, which is the simplest amino acid that we have here. In amino acids that we have a carboxyl group, amine group, and only a hydrogen, H. So glycine is written as glycine, shown by GLY, and the letter for glycine is G. It is important to memorize these letters and how they are shown in how they're shown, because like imagine you have like a chain of, let's say, 1000 amino acid. Is it easier to write down glycine, alanine, valine, and so on, or is it easier to just write GAV? That's gonna be four letters only, and then you would have written your whole protein structure. So this is the glycine. Now let's move on to the next one, the next nonpolar amino acid that we have here. As we said, all amino acids have the exact same structure. The only thing that's differ is your side chain. So we're gonna have the same exact structure here. We're gonna remove the side chain and we're gonna put a methyl group there, and we're gonna call it alanine. We're gonna show it with ALA and with a letter A. This is basically the alanine amino acid. It's another nonpolar amino acid. The other one that we're gonna have is called, again, same exact structure as before. We're gonna put a CH2 here. We're gonna, CH1, CH. We're gonna add another methyl here, and then another methyl right here. We're gonna call this valine. So now we're gonna have all 7 amino acids written. So now here's all 7 nonpolar amino acids that we talk about. Among them, again, glycine is the smallest, and proline here, the amino acid right here, is unusual because its side chains here, it loops back and bonds to its own amino acid group, forming a ring structure. And that structural quirk matters later when we talk about the protein folding, because proline physically cannot bend the way other amino acids do. So when we talk about the alpha helices, we're gonna say that one thing that won't form that is not really favorable for forming alpha helices are proline and glycine, and then we're gonna talk about them later, but these are the amino acids that you should know. Then this is the first category of the 7 out of 20 amino acids that we talk about. Then we're going to talk about the aromatic amino acids. The aromatic amino acids are phenylalanine. As we said before, they have the same structure except for their side chain. So the phenylalanine, the first aromatic that we're going to talk about, looks something like this. There's a CH2, and there is a ring structure, benzene ring here. And we're going to say that. And this is called phenylalanine. We're going to show it with PHE, and then we're going to say F, the letter F. So now we're going to talk about the other one, which is tyrosine. The difference between the tyrosine and the phenylalanine is only an OH group here. I'm sorry for an OH group here. That's going to be tyrosine. We're going to show it with TYR, and then the letter is T. The next one is really different is called tryptophan. The tryptophan is, sorry, I'll show it here. The difference between the tryptophan is it's not a benzene ring anymore. There are two ring structure here. One is, let me draw it like this, an NH group here. It's gonna be easier if I write it down. An NH group here. With its ring structure, with a benzene ring here. This is called tryptophan. We're going to show it with TRP and the letter W. These are the aromatic that we have, aromatic amino acids. Then the next group is the polar. Here's another catch that might be useful. The aromatic amino acids absorb UV light in 270 to 280 nanometers. 270 to 280 nanometers, and this is important when you are in the lab trying to understand how the protein concentration, and we're going to talk about it later, but this is how you measure the protein concentration in the lab. Then we're going to talk about the polar uncharged amino acids. We have 5 of them. The first one, the most important one basically, kind of, is cysteine, but we're gonna follow the same pattern as so the difference between the serine, which is kind of an easy amino acid to draw, the only thing is we have an OH group here, we have a CH2 group here and OH here. This is called serine, and we're gonna show it with SER, the letter S. We're gonna have cysteine. We're gonna have cysteine here. Cysteine is kind of special because, as you probably know, we form disulfide bonds, and the reason is because we have an SH group here, and then in a pH value, different pH values, we're gonna lose this hydrogen, and we're gonna gain charges, and then we're gonna form disulfide bonds. And we're gonna talk about it later, but this is the important thing that we need to know about the serine. And we're gonna cysteine. I'm sorry, cysteine. Basically, when we form a disulfide bond, we're not, with 2 cysteine amino acids, we're not going to call it cysteine anymore. We'll call it cysteine. So it is important to know that the difference between the cysteine and cysteine. But we're going to talk about it later anyways. These are called polar uncharged amino acids. Now the next group is therionin. I'm gonna put it right here, and basically the difference between the serine and therionin is just basically that there is one extra methyl group to it. So we're gonna draw the methyl group here and another OH group here, and this is called therionin. We're going to show it with THR-T. The next group is so these are the polar uncharged group amino acids. We're going to have the asparagine, and we're going to have glutamine here. I'm going to show these because these are kind of big, almost. The asparagine is, but they also contain that OH group. Again, this is the asparagine, CH2, C, double bond, and another NH2. We call this sparginin. We're going to show it with ASN with the letter N. Next, we're going to have glutamine. Same thing, almost, with an extra CH2. CH2 here. And these are called the polar uncharged amino acids. We have 5 of them. Have side chains. If you look at them, they have side chains that can form hydrogen bonds, but they don't carry a physiological charge they don't carry a charge at physiological pH. And one of them, the cysteine that we talk about here, deserves a flag, basically, disulfide bonds, as we talk about. Essentially stitching 2 parts of a protein together. They're huge for structure, and we'll build on to it in the next episode, as we said. Now, we talk about the polar uncharged amino acids. Let's say, okay, we had 7 nonpolar amino acids here. Nonpolar. We have 7 of these. We have 3 aromatics, 10 by now. Then we talk about 5 polar uncharged amino acids here. 15 of them now. So now we're going to talk about the positively charged amino acids, and we're going to talk about the negatively charged amino acids. The positively charged amino acids are basically the basic ones. And I'm going to stop the video here because it's going to make so it's been a long time that I've just I don't want to waste time on drawing the amino acid. It was just like a way to show you guys that here it is. We have the same structure, we have the same building blocks. We're only changing the side chain. So I'm gonna draw the charged amino acids, and we're gonna talk about them. So now I drew the 5 charged amino acids that we have. 3 of them, 3 above, are the positively charged, and the 2 below are the negatively charged amino acids. As we can see, the first one is lysine. We have an Na3 plus. That plus is what gives it the positive charge. Again, for arginine, which is shown as letter R and ARG. And then we have histidine with an H, and there's a ring structure here. It's not aromatic. There's a ring here. Again, we have the next 2 negatively charged, aspartic acid and glutamic acid. And if you look at it, they look similar, but the only difference is that the glutamic acid have 1 more CH2 group in it. And basically, these are the only thing that we need to know about the structure and the charges of the amino acids. But the important thing, 10 of these 20 show constantly enough that you should be able to draw them from memory and recognize them on side. It's called, let's go from the very beginning. We need to know, should I erase this? Sorry for that. So, the 10 amino acids that we need to know is called, glycine is important, alanine is important, valine is important, and in here we're gonna need to know about the arginine. Oh, sorry. Right. Yeah. Sorry. Arginine is important. Lysine is important. Histidine is important because these 5 are important. The reason is these are the ionizable group, and these have basically later on when we're talking about the phosphoelectric, the PI, which is the phosphoelectric point. It is really important to know about the ionizable groups. Then we need to know about the cysteine as well and the serine as well, because these are the mostly shown, and then we need to know about the OH group, and then we need to know that they're the polarizable. So we talk about amino acids enough. Let's go about the chirality. So we're going to talk about why shape matters. So look at let me make this bigger. So we have an alpha carbon here, and if you look at it again, it's bonded to 4 different groups. We have arranged in 3D space around the central point, and anytime a carbon has 4 different substituents like that, it's called chiral center. So a characteristic of a chiral is that it needs to be bonded to 4 different groups or atoms, and it means molecule can exist in 2 mirror image versions that are not superimposable on each other, which would be this one and the other one. Look at your hand. They're like they look like in mirror images, but they won't be superimposable if you want to put them on top of each other. You're not gonna have it exactly. Your thumbs would be on the other side. So for example, if you want to put your right hand on your left hand, your thumb would direct to the left, and for your left hand thumb would be directed to the right. So you see, it's not superimposable, and these are called chiral centers. And no matter how you rotate them, you can't lay one perfectly on top of the other one. So these mirror image pairs are called enantiomers. An amino acid come in 2 forms. We have the L form, and then we have the D form. Here's a key fact that MCAT wants you to know, to walk away with. In a biological system, the L form, this one, is what actually get used. And why? Because the enzymes that build amino acids in your cells are themselves chiral and only recognize and produce one specific version. Biologically, biology isn't picking L at random. It's a direct consequence of enzyme specificity. And one more detail worth knowing is that glycine is a single exception to all of this. Its side chain is just a hydrogen. Which means let me go to where the glycine was. I want to show you this. Which basically means that the alpha carbon is bounded to 2 identical hydrogen and no 4 different groups. Now, chirality, glycine is achiral and the only one of the 20 amino acids. See, 1, 2. We have H, we have COH, carboxyl, and we have amine group. We don't have 4 different we don't have 4 different atoms. See, H, methyl, COH, and H2, 4. But here we have 3. So glycine is achiral, and this is the thing for the chirality. Now, the important part is the ionization of amino acids that we're going to talk about. Let me draw an atom first, like an amino acid first. We're going to draw it in this way. I'm going to make it bigger. An O group here, an O group here, negative charge, we have the NH3 plus here. We're gonna have the R group, H. Let me make this bigger. Perfect. So this is our amino acid, and we're going to talk about the ionization of amino acid. The carboxyl and amino group can act as an acid or base. This is kind of obvious from the charges that we have here. Molecules that can be a base or an acid simultaneously are called amphoteric. At physiological pH 72, the carboxyl group donates, this one, the COH, it donates a hydrogen, while the amine group accepts that hydrogen, the NH2. I'm going to show it like this. This one is better. The NH2 at physiological pH 72 accepts the hydrogen. It gains an extra positive charge, so it would be positive. The COOH, the carboxyl group, loses that hydrogen. It loses one positive charge, so it would become -1. +1. And at pH 7.2, amino acids are dipolar ions, and they are called zwitterions. And despite charges at pH 7.2, these amino acids are neutral. Why? Because we have one negative and then we have one positive. So we're gonna get a neutral atom here. Now, here's the important thing that you need to know about the amino acids, that's zwitterions and aliphatic molecules. Let's talk about a non-ionic form of amino acid here. We're gonna go with, let me draw an amino acid here first. H, R. To make it easier, I'm gonna put R here. NH2, COOH. This is a non-ionic form. Non-ionic form. At pH 7.2, I'm gonna draw it here. R, carboxylic and amine group. At pH 7.2, carboxyl. I'm gonna say COH donates proton or deprotonated. Amine group accepts proton or protonated. Overall, no net charge. So this is the first condition that we have. The second one, I'm going to use this one as a template. The second condition that we're going to talk about is when we are at the high pH. So, I'm sorry for that one. This is CO negative, positive. I'm really sorry. 3 plus. We are looking at a high pH here. Not gonna do anything about the numbers. So, if you have a zwitterion as acid here, so this is a reversible reaction. At high pH, we have lots of OH group. So the OH is negatively charged. We have lots of OH here. Here's what happens. Do you think when we have a lot of OH in an environment, so I'm looking for here, and NH2 here. NH2 accepts H+. Carboxylic group loses proton, sorry for that, loses proton. This one accepts proton, and then we're in an environment with a negatively charged that we want proton. So what do you think, which group would get ionized? Which protein loses its hydrogen? Is it the amine group or carboxylic group? It's a carboxylic group. So the amine group would not lose any proton, while the carboxylic group loses its proton and it gains a negative charge, and then we're gonna lose that proton. So at high pH above 7.2, the H+ is lost from NH3 plus. That's one. We're gonna lose the proton here as well. But this is the ground form that we're gonna have. This is NH2. We're not gonna go for NH1. NH3 plus becomes neutral, and this is how we lose that proton here. So the overall charge is negative now, because this is the only charge that we have here. Now we're gonna go for the low pH conditions. Sorry, we're gonna have an arrow here and then C R. Oh, okay. I'm not going to show any charges yet. At low pH, below 7.2. Here's the thing. At low pH means it's highly acidic. We have lots and lots of, I'm going to show it here. We have lots of H plus in the environment. So do you think the NH2 that we have, where would the proton group, would they try to bind to the NH3 plus or the carboxylic group here? Obviously they would attach to the carboxylic group. So the carboxylic group would gain one proton here, and then the only group left now is that one, the positive charge and the amine group. And then so basically at low pH, H plus, the proton is gained by the carboxylic group. The carboxylic group becomes neutral. The overall charge is positive. And so that's basically how the amino acids react in a different pH levels. So let's have a recap and let's bring it together. Every amino acid share one backbone. Let's have a recap. Amino acids shares one backbone. We have amine group, this one. We have the carboxylic group, we have the hydrogen, and then we have the side chain here. And the only thing that changes is the OR group. So an amino acid, that's the only thing that's a variable group that we have. The OR group sorts all 20 amino acids into 5 classes of nonpolar, and then the aromatic, polar uncharged, and positively and negatively charged here. Most amino acids are chiral, except for the glycine, and existing as L and D enantiomers, with biology almost exclusively using the L form because of the enzymes. Again, except for glycine, which has no chirality at all, and their charge isn't fixed. It shifts with pH. Sitting neutral as a zwitterion at physiological pH, going positive in acid and going negative in base. So you're not memorizing 20 separate molecules anymore. You're applying one framework 20 times. In the next video, we take these individual amino acids and start linking them together through the peptide bond, where the ionization behavior gets even more interesting once amino acids are locked into a chain. So subscribe, you don't miss it. And if this helped clear things up, drop a like. It genuinely helps the series reach more students studying for the same exam